Asha, a cute baby, slowly started to experience a loss of muscle movement. She was soon diagnosed with spinal muscular atrophy, a rare autosomal recessive genetic disorder that destroys motor neurons in spinal cord and brain stem, leading to loss of muscle movement like breathing, swallowing, and ultimately death before the age of 2. The cause lies in loss of SMN1 gene, which normally produces the essential SMN protein for motor neuron survival. Early treatment like nusinersen Spinraza worked by boosting the backup SMN2 gene to produce more SMN protein, but these required repeated lifelong administrations and only managed the symptoms, not the root cause. This is where Zolgensma enters the story. This groundbreaking gene therapy was approved by FDA in 2019 and provides a new working copy of SMN1 gene with a single dose IV infusion, thus helping SMA patients like Asha lead a happy and a healthy life. Let's first travel back in time and explore the amazing journey that provides the base for this discovery. The story begins when scientists first discovered that DNA is the one which carries genetic information and worked to understand the genetic code. This led to development of rDNA technology and ultimately gene therapy. The first successful gene therapy trial was conducted in 1990 on a little girl with ADA deficiency. However, in 1999, this field saw a major setback when a patient died due to severe inflammatory response to adenovirus vector. This incident led to major pause in gene therapy trials, stricter safety rules, and better protection to make it safer. Over next 2 decades, the researchers worked on making it safer and develop more effective vectors. However, developing a gene therapy for SMA was complex. The major hurdle was the blood-brain barrier, a highly selective membrane that acts as the shield for CNS, preventing harmful substances, even therapeutic molecules, from entering. So, for many years, gene therapies targeting CNS had to be administered via highly invasive direct injections into spinal fluid or brain, and this was highly risky for infants. Soon, the discovery of AAV9 vector revolutionized gene therapy for treating neurological diseases like SMA. Isolated from primate tissues in early 2000, it showed unique capacity to safely cross the blood-brain barrier and made it possible to develop an approved one-time gene therapy like Zolgensma. Here the functional SMN1 copy of the gene is delivered using AAV9 vector to the target CNS motor neurons, and now this SMN1 gene produces the SMN protein which was earlier deficient in these patients. This method demonstrated success in restoring the production of SMN protein in infants. This Zolgensma also made headlines on being the most expensive drug, around $2.1 million per dose, raising ethical and economic debate over its accessibility, affordability, and also setting pricing standard for gene therapy. Zolgensma also has some side effects such as elevation in liver enzymes, rare cardiac issues which makes monitoring and corticosteroid use highly necessary. Zolgensma is most effective when given early as it cannot reverse the damage caused by SMA. Despite this, Zolgensma has been a major breakthrough, being the first approved gene therapy for SMA. And also, the success of Zolgensma has sparked interest in investigating gene therapies for other neurological disorders. Currently, it is under clinical trials to treat older patients.